Can you hear me?
Yeah, I can see you. I can hear you, yeah.
Okay.
Okay, please.
Hi everyone. My name is Tongda Xu from Fujian Agricultural and Forestry University in China. First, I'd like to thank organizers and committees to give me this great opportunity to show my recent work progresses on auxin signaling. My lab has been worked on TMK-based auxin signaling on plasma membrane for more than 10 years. Sorry. As we all know that auxin has an astonishing and diverse functions in plant development, including regulating primary and later root development, embryogenesis, and also organogenesis. And in the meanwhile, the function of auxin is also really complicated. Sometimes you will see it promote growth, but also sometimes it inhibits growth. In general, we know that low concentration of auxin seem to promote growth, while high concentration of auxin started to inhibit growth. And at the different organs showed us different sensitivity to auxin, like the root is very sensitive to auxin and the stems is less sensitive to auxin. So the function of auxin seems to be dependent on its auxin concentrations. Why its concentration of auxin can be interpreted to different downstream signaling that can regulate cell elongation, cell divisions, and cell differentiations, which further regulate plant development. So we all know that there is a well-established auxin signaling in nucleus, which is mediated by its receptor TL1, which is a key component. In E3 ligase complex. Auxin promotes the interaction between the TL1 and the auxin, which is lead to the ubiquitination and further protease-dependent protein degradation. And the auxin protein actually is a transcriptional repressors, and its degradation by auxin will release ARF transcription factors and then trigger the auxin gene expression. So here is the transcriptional auxin signaling. However, by the recent technical advances of proteomics, we found that short-time auxin treatment will induce a global protein phosphorylation in plants. So we use different concentration of auxin treatment, which show that high auxin can induce more protein phosphorylation events in plants. This suggests another level of auxin regulation in plants other than the TL1-based transcriptional regulations. And my lab focused on receptor-like kinase called TMK on plasma membrane that play essential roles in protein phosphorylation-based auxin signaling. There are 4 members of TMKs in plant Abdopsias genome, that show the redundant function in regulating both plant development and auxin responses. You can see here that the TMK14 double mutant already show the severe growth phenotype, and the quadruple mutant is sibling lethal. You will see this very abnormal growth patterns in this mutant. Thus, we propose there exists another auxin signaling cascade that might be triggered on the plasma membrane, as we know that auxin can be transported to the extracellular domains, extracellular regions, which might be perceived by this signaling systems. So then we started to investigate deeply how this pathway is triggered and transduced in the plant development. The first key question is how auxin is perceived for this TMK-based auxin signaling transductions. So previously, we found that auxin can stimulate complex formation of ABP1 and auxin binding protein with TMK on the plasma membrane, which is dependent on auxin concentrations. Thus, we propose that ABP1 might be required for auxin perception on plasma membrane. However, later on, another study showed that ABP1 CRISPR mutant showed a similar growth and auxin response as in wild type, which put ABP1's function in debate for its function in both auxin responses and plant development. And the previous ABP1 knockout mutant, which showed embryo-lethal phenotype, actually is caused by the silencing of the adjacent genes. This put ABP1's function in question again. And we previously used a point mutation line called ABP1-5, which is a tilling line from another lab, also showed a complex mutations in genome background. Thus, we generate a new ABP1-5 mutant by directly transformed mutant ABP1-5 protein to the ABP1 knockout mutant. So this is like the clean ABP1-5 mutant. And the new ABP1-5 mutant shows severe growth defects, like this abnormal cotyledons, the a single cotyledon or triple cotyledon or fused cotyledon, which is shown as in the TMK mutants. And regarding auxin responses, we use a hypocaudal elongation. You can see that in wild type, auxin can promote hypocaudal elongations. But in this ABP1-5 mutant, you can see that it's totally insensitive to auxin. This suggests that auxin binding pocket ABP1 is important for the either auxin signaling or plant development. So this controversial of ABP1 is that its knockout mutant has no obvious phenotype, but the even biochemistry data or the point mutation mutant or the antisense or antibody lines showed a strong phenotype. So how to experience this phenomenon? The most possible reason might be the functional redundancy, but it depends on the sequence similarity. We did not find the ABP1 homologs in abducted genome. So how to explain this? But when we look at the structure of ABP1, we found that ABP1 is mostly a structure-based proteins which do not have any functional domains. Thus, if we propose that if there is a protein that has similar structure of ABP1, it might function similar to ABP1. So if this is true, this protein might also have the binding affinity to TMK as ABP1. Thus, we use the TMK to find out the potential ABP1-like proteins. And by powerful mass spectral analysis to detect the TMK interaction proteins, we are lucky to find a protein which belongs to the same family of ABP1 can bind with TMK specifically. We name it ABP1-like one. Based on the structural simulation through AlphaFold systems, we found that ABL1 have the similar structure as ABP1, especially the co-region for auxin binding pocket. You see the red is auxin, the blue is the zinc iron. And based on the sequence analysis, we also identify other two ABP1-like proteins. We named ABL2, ABL3. We can see that the amino acids that are required for auxin binding are very conserved in these ABP1-like proteins. And also we use immunogold to detect that ABP1-like protein actually localized to apoplast as a secretive proteins that provide us a special possibility to bind with extracellular domains like TMKs. By different methods, actually, we found that auxin can promote the complex formation for ABL proteins with TMK, which is similar to the ABP1. You can see here, both ABL1 and ABL2 can bind with TMK in auxin-dependent manners. And then we mostly tested the genetic relationship between ABL1 protein and the ABP1 mutant. Here we can see that the single mutant for ABP1, ABL1, ABL2 actually do not have an obvious phenotype. But the double mutant for ABL1, ABL2 began to show severe growth phenotype, including the smaller size of the plant and the curling leaves. And when we introduce ABP1 knockout mutant to this double mutant, you can generate a triple mutant shows a severe growth pattern, severe growth defects and the double mutant. This suggests ABP1 might have the functional redundancy for the ABP1-like proteins. And this phenotype can fully rescued by the ABL1, but partially rescued by the ABP1. And interesting, ABP1 cannot rescue this curling leaf phenotypes. This suggests that ABP1-like proteins can ABP1 have redundant function, but also have distinctive functions. And this phenotype cannot be rescued as a point mutant mutation version of the ABL1, ABL1, M2, and ABP1-5. The mutation happened in the auxin binding pocket. So this mutated protein cannot rescue phenotype. And then we test whether ABP1 and the ABP1-like fungal redundant through TMK by introduce both ABP1 and ABL1 mutant to the TMK14 heterozygous. Heterozygous has showed a weaker phenotype. You can see that either the single knockout mutant of ABP1 or ABL1 cannot strongly enhance the phenotype, but the double mutant strongly enhance the TMK14 double mutant phenotype. This quadruple mutant shows a very severe growth defects. And this phenotype can partially rescued either by the ABP1 or ABL1. And also we test the auxin responses to see the similar pattern. This suggests that the ABL1 and the ABP1 function through TMKs in both plant development and auxin responses. And then we asked whether ABP1 like can bind the auxin directly. By the MST method, we found that both ABP1 and ABL1 can bind it with the auxins with a micromole KD, and the point mutation, the ABP1-5 or ABL1M2, you see the binding was totally lost. And more interesting, we see that TMK1 extracellular domain itself has the auxin binding affinity, which is similar to the ABP1 or ABL1. And when we mix the ABP1 or ABL1 with TMK extracellular domain, it will strongly enhance the auxin binding affinities. So this strongly suggests that ABP1, ABP1-like, and TMK might form a co-receptor system for auxin on the plasma membrane. Thus, we propose an extracellular auxin perception systems by the co-receptor of ABP1, ABL1, and TMK on plasma membrane. And this work actually will put an end for the debate of ABP1 functions in plant development and auxin signaling. And we also show that an interesting gene composition by a structure-similar protein, not by sequence-similar proteins. And this work is currently under the revision status. Then I will introduce how auxin signal is transduced by TMK systems. As a receptor kinase, not surprisingly, TMK is localized to the plasma membranes in different cells, including the leaf cells, root cells. On the plasma membrane, as a kinase, auxin can stimulate the TMK interaction with AHA, which is a proton transporters, and which can activate the AHA and promote the proton export and exudation of the cell wall and promote the cell expansions. So this work actually nicely explains how auxin promote acid growth regarding the cell expansion. And recently, we also found that TMK can directly interact with PIN proteins, auxin transporters that determine PIN localizations. You see that in the mutant, they're actually dislocalized. And this will provide a self-regulatory mechanism for auxin signaling and auxin transport itself, which has been reported for a long time, but the mechanism is unknown when we explore the mechanisms. So it's easy to understand the function of TMK on plasma membrane and the function as a kinase to phosphorate its substrate. But occasionally, actually, we can see TMK localized to the nucleus. At the beginning, we thought it is an artifact. And then my student also found that auxin treatment can stimulate a formation of a short band for TMK. By mass spectrum, we found that this is actually intracellular domain of a kinase domain of TMK. And then we realized that auxin might promote the cleavage of TMK, which might be translocated to the cytosol and the nucleus. So then we ask why TMK is cleaved and when TMK is cleaved. We found a nice system called apical hook development. At the maintenance stage of the hook, actually auxin is accumulated at the concave side or inner side of the hook. In this specific stages, you can see TMK is cleaved. And also we can see the TMK localization to the nucleus in these specific stages. This suggests that auxin, TMK is cleaved at concave site where auxin accumulates. And we know that in the wild type, actual cell elongation was in the inner side of hook is inhibited, and at the outer side of the hook is promoted. And this will cause the differential growth from the inner to the outer side, which cause the bendings. And the increased auxin levels in UCAR1D mutants cause both side of inhibitions. And in the TA mutant, in the low auxin actually release both side of inhibitions. Both will cause the opening of the hook. So this is indicate the differential growth is important. And also we can see that the auxin actually inhibits cell elongation in these regions. And we also found that TMK single mutant displayed apical hook development effects at the maintenance stage. You can see the hook will pre-opened. And this phenotype is similar to auxin biosynthesis defective mutant. And the growth inhibition at the inner side of the hook is abolished in the TMK1 mutant, and is also showed insensitiveness to the auxin. Then we try to identify the potential interaction proteins for TMK1 cleavage product. We are surprised to see that specific auxin proteins, I32 and 34, specifically interact with the TMKs. And which is specificity for the I32 and 34. Actually, they belong to a family of non-canonical auxin proteins, which lack a proper domain to which is required for T1 interactions. So like the I32 and 34 actually cannot be recognized by T1, but specifically recognized by the TMKs. And by the fusion of the promoter-driven auxin 32, we found that I32 is highly accumulated the hook maintenance stage when auxin accumulates and when TMK cleavage occurred. More interestingly, opposite to the auxin degradation of auxin degrades the canonical auxin through T1 pathway, you can see that actually auxin promotes the protein stability of the both I32 and 34 in a TMK1 dependent manners. So finally, we propose that the differential auxin responses happen at the two sides of the apical hook. It seems to us that auxin accumulate at the concave side of the apical hook to cleave the TMK and then stabilize auxin 32 and 34, and which inhibits gene transcription and required for the local growth inhibitions. So this suggests that there's TMK's function might have different on the plasma membrane and also cleaved into the nucleus and the cytosol, because we know that on plasma membrane auxin TMK can activate AHA to promote growth, but it seems that after cleaved, it starts to inhibit. To further investigate whether this is true, we identify the cleavage site of TMK. Actually, it's is an adenine amino acid, 527th amino acid, which is just after the transmembrane domain of the TMK. You see that by mutation of this cleavage site, it will totally block the auxin TMK cleavage, TMK1 cleavage in plants. And also we see that auxin cannot promote a cleavage if we mutate this site. So then we use Hapkato as a system to start cell elongations. We found that you can see if we overexpression the TMK1C C terminals, it will inhibit Hapkato elongations like here significantly. But when we use the point mutation lines, which cannot be cleaved, we found that it started to obtain a very strong ability to promote growth, even so the ability is stronger than the Y-type TMK. Like in here, the point mutation will enhance the Hapkato elongations and the Y-type in the TMK1 double mutant background. And not only for the Hapkato elongation, we also found that the TMK non-cleaved version can generate promote growth, and the cleaved version, like a TMK C terminal, can generate inhibit growth. Like in here, that's root, we can see that promoted by the non-cleaved version, and the C terminal can inhibit roots. And the plant size also shows similar patterns. You can see that the non-cleaved version can promote growth, and the cleaved version can inhibit growth. This is like the general mechanisms for promote or inhibit growth. This is mediated by the cleavage. As we all know that auxin can promote the TMK1 cleavage. So then we test the function of TMK cleavage in auxin responses. We knew that short auxin treatment can stimulate Hapkato elongation in dark. And when we introduce the TMK point mutation, which cannot cleave, you can see it can fully rescue the defects in the TMK14 double mutant about auxin responses. But we know that a long-time treatment can start to inhibit Hapkato elongations in wild types. But if we introduce these point mutation lines, that TMK1 cannot be cleaved, and you can see this growth inhibition by the auxin is strongly reduced in these point mutation lines. This suggests that TMK cleavage is required for the function of auxin to inhibit half-cut elongations. So we propose a model that TMK4 length and the TMK C terminal have the opposite function regarding promote and inhibit growth. And auxin-mediated TMK cleavage is like the function as a molecular switch to switch on the function about from the promote growth to the inhibit growth. The concentration effects of auxin actually very interesting to us, and this is not only happened in the cell levels or tissue levels, but also happened in the species levels. Likely in the monocot plants, like the rice is less sensitive to auxin, and the dicot plants like the tobacco is more sensitive to auxin. And we can see that the same concentration of the auxin can promote growth in rice, but inhibit growth in tobaccos. So we are currently trying to figure out whether the underlying mechanism is also dependent on the TMK1 cleavage. So overall, actually, we figure out how extracellular auxin work from the perceptions on the plasma membrane and how auxin is transduced from this ABP1 and the TMK systems. And we also figure out plasma membrane-based TMK function and also cleavage-based function, which they have the totally opposite function regarding plant growth. And this also explains the interesting phenomena of auxin in plant growth. Finally, I would like to thank all my lab members, especially Yong Qiang, actually, who's working on ABP1 live proteins, which is a very difficult project. And also Cao Ming and Chen Rong actually working on the TMK1 cleavage, which is published on Nature already. And currently we are submitting a paper that regarding TMK1 regulate pink proteins and also TMK1, how TMK1 cleavage is regulated. And also collaborators, Zheng Biao, actually my previous supervisor, we collaborating on ABP1 like proteins, like stories, which is I think is important for the auxin field. And we collaborate with Yuji Freeman for pink stories, and Jiang Liwen from the Hong Kong actually helped with the immunogold staining for ABP1 like proteins, and Jian Feng actually helped with the TMK1 cleavage. And we thank all funding support from China. Okay. Thanks, and we are ready to the question. I'm ready for the questions.
Thank you, Dr. Xu, for sharing the interesting work on auxin signaling. Now let's start the section of Q&A and questions from the committee members. Please turn on the microphone.
Hi, Tongla.
Hey.
It's a great talk. Just one short question for TMK cleavage. Whether you test the cleavage depend on ABP and ABPL like protein?
Yes, actually, we tested by the both ABP1 and ABP1-5. One is functional and one is the unfunctional. We show that actually ABP1 can promote cleavage, but ABP1-5 cannot. So this is also indicates that the cleavage is depending on the ABP1.
Mm-hmm. Yeah, that's interesting. Yeah. Also, another question: did you find there which kind of enzyme can precise the TMK?
Yes, actually, we have finished this work submitting. Okay. Actually, it's a meta-caspase can specifically cleave TMK1 regarding the in response to auxins. Yeah.
Okay. Okay. Thank you.
Identify them as protease. Mm-hmm.
Now really impressive data, nice presentation. So I'm just wondering, I remember ABP has been described or has been mentioned as an ancient protein present in algae. Mm-hmm. Is this mm-hmm. true for the ABP likes? And what is then this apoplastic auxin signaling mainly doing? Is it a kind of general function that is is present already in unicellular systems, in algae and so on, and is mainly related to, let's say, cell elongation without doing much more, and that then the nuclear auxin responses and more elaborated and more for specific developmental processes. Because looking to all the phenotypes that you have been discussing, they are really very basic. Cell elongation, taking the apical hook, hypocotyl elongation, root elongation. It's not really a kind of very specific developmental function or aberrant phenotypes that needs to have or are relying on a very specific signaling cascade. It's really very basic, what you call grow. And is this something that's an ancient conserved auxin signaling that is already present in unicellular systems?
Yeah, actually, this is a very good section, very good questions. And like in green algae, actually, that both ABP1 and ABP1 already exist, and TMK is already exist, but TL1 is not exist. So this suggests that this signaling pathway might be more ancient than the TL1 pathway, because in the like the single cell systems, it is only involved in the cell elongation and the cell divisions, not too complicated. So maybe this pathway already can facilitate both regulating the cell elongation or cell divisions. Actually, we have another work actually is still in progress that after auxin is increased concentrations, actually it will trigger the cell division through the TMK dependent pathway and the TMK cleavage. And this might be the, I think it's maybe fit to this kind of single cell systems.
And in the first, the representative of the first land plant. So if you look into the mosses, like in Physcomitrium, and so you have this ABPL family also there, or it's much more reduced?
Yeah, they already have ABP1-like families there, and we also identify very ancient ABP1-like, which is similar to the ABP1. We call it ABP1-15 in the Abdopsis. Currently, we are working on that ABP1-like to see what's the function of this ABP1-like in Abdopsis. Yeah.
Okay, thank you.
Okay, Tumda, I have a question. So, is there any crosstalk between the ABP1 TMK1 pathway and the TAL1 mediated auxin nuclear signaler pathway? This is first question. Second question is from the evolutionary perspective. So, which pathway is appear first in the evolution of plants?
Actually, answer the second question first. Actually, I already said that in the green algae, actually only the ABP1 and ABP1-like exist, but the TL1 is not exist. So this means that maybe this pathway appear first, and the second path
actually compare with the TL1 mediated nuclear signaling pathway with your TMK1 pathway, which one appear first in the evolution?
ABP1 appear first in the evolution.
Okay, thank you.
It's very clear in the green algae. And definitely there's crosstalk between this ABP1 and the TMK with the TL1 pathway. Actually, we see that the majority of the components of TMK pathway the expression is depending on the TL1 pathway. And of course, this crosstalk is very actually decent and complicated. And we are trying to figure out how this kind of coordination happens in response to a certain concentration of auxin. But this is very complicated and still need time to figure out.
Okay, thank you. Thank you very much.